Hello and welcome to presentation 4 of 5 of an Introduction to Genetics for the Alberta Science 30 curriculum. This covers topics in Unit 2 of Biology. Let's talk a little bit about sex linkage. Gender and inheritance experiments was started through the work of Thomas Hunt Morgan, an American geneticist who worked in the early 1900s on the fruit fly or Drosophila melanogaster, which provided a deeper understanding of heredity based on gender. He chose Drosophila because of several things. They have a rapid lifestyle or cycle. They're only alive between 10 to 15 days. They lay hundreds of eggs at a time. You can do crossing experiments many times. Offspring mature quickly. You can tell the difference between males and females. Females are larger, and males have that black colored abdomen. They are small. They have contrasting traits. They only have 8 chromosomes or 4 pairs of chromosomes. Morgan found the following through analysis. Females had 4 homologous pairs of chromosomes, which consisted of 3 autosomal pairs and 1 sex pair, so it had an XX. Males only had 3 homologous pairs of chromosomes. They had 3 autosomal pairs, but the sex chromosomes were not homologous. Like humans, they had 1 X and 1 Y chromosome. He looked at eye color in Drosophila. So red eye color was dominant. Red is represented by big R and white is represented by little r. Males were mostly white and females were mostly red. He assumed that these alleles were found on the X chromosome only. So let's look at 2 crosses. One is a cross of a red-eyed female with a white-eyed male. The other is a cross of a red-eye, white-eye female with a red-eyed male. In your first cross, you have the X chromosome with the white-eyed allele and a Y chromosome with a female that is got a red-eyed allele on the X chromosome and another red-eyed allele on the X chromosome. When you do these crosses, you get 4 offspring produced. The 2 females produced are heterozygous for the red-eyed allele. The 2 males are red-eyed. All of your offspring are red-eyed. That's their phenotype. In the second generation, or the F1 cross of that same cross, you have a red-eyed male crossed with a heterozygote. Your result is 1 female that is homozygous for red eyes, 1 female that is heterozygous and displays red eyes, 1 red-eyed male and 1 white-eyed male. You get 3 red offspring and 1 white male. The genes controlling eye color must therefore be on the X chromosome. Y does not carry the gene for eye color. Males cannot be heterozygous for sex-linked traits. They can only contain 1 allele. Your female choice, or the possibilities for females are homozygous dominant, homozygous dominant, homozygous recessive, and heterozygous, or containing 1 red allele, 1 white allele. Males can only either contain a white allele or a red allele. Traits controlled by genes carried on the Y chromosome only affect males. Sex-linked traits are controlled by genes that are located on the sex chromosomes. Humans have 22 pairs of autosomes and 1 pair of sex chromosomes. Males are represented by XY and females are represented by XX. If you'll notice, the X chromosome is significantly larger than the Y chromosome. Sex-linked linked traits are almost always on the X chromosome. The Y chromosome is used primarily for sex determination, or indicates or determines that the individual is a male. It occurs in males, more males than females. Why? Because the males only have one X chromosome, so they are more impacted by changes to the X chromosome than females are. So a cross of a red-eyed female, we go back to the Drosophila cross, so a heterozygous red-eyed female with a white-eyed male. So we have white eye X, Y, red allele X, white allele X. You have a red-eyed female produced, a white-eyed female produced, a red-eyed female produced, or red-eyed male produced, and a white-eyed male produced. So you have one red female, one red male, one white female, one white male. Those are your phenotypes. What are some examples of sex linkage in humans? If you are not colorblind, red-green colorblind, you will see that 5 in green in those dots. Color blindness is a sex-linked trait. The genes for visual pigments that are responsible for perception of red and green are found on the X chromosome. Can you see what this number is? If you were red-green colorblind, you couldn't. Female genotypes. XC is considered to C is color blindness gene. So XC says that you can see color, normal female. Big C, little c means you're a carrier. You're carrying the color blindness gene. Little c is a colorblind female. The phenotypes for females are normal or colorblind. Will a carrier be colorblind? No, because it contains a dominant allele. The male genotypes can either be big CY, or a normal male, or little cy, a colorblind male. The phenotypes again can be normal and colorblind. Males cannot be carriers. They either contain the dominant or recessive allele, but they cannot have both dominant and recessive because it's an X-linked trait. So here's what red-green colorblind would look like to somebody affected. Hemophilia is the trouble with clotting blood. It's caused by the lack of a blood protein called factor VIII, which is critical for blood clotting. The gene for factor VIII is located on the X chromosome. It occurs more in males than females. Affected males are homozygous recessive for the affected gene. Carriers are heterozygous, contain one dominant and one recessive allele, and boys who are affected contain only the recessive allele. Not affected males contain only the dominant allele. Sex-limited traits are only expressed in one gender. For example, milk production in cows is sex-limited to females and it's controlled by a gene. Bulls carry these genes but do not make milk, so they do not express the gene. So here's an example of a bull. So limited means it occurs in one gender and not the other, even though both genders carry the gene. Sex-influenced mean that it's more common in one gender than the other. For example, baldness is influenced by the male hormone testosterone. So you'll see more men going bald than women, but women can also go bald. The gene is dominant in males and recessive in females. So baldness is more common in men than in women.